Hey guys, first things first, I would like to apologize for I only have my phone to record my presentation and to create my PowerPoint. So please forgive me. Moving on, today I'm going to present about my critique on a thesis naming marine biofilm evading eukaryotic predation by the use of chemical defenses. This thesis was constructed by these critical thinkers, namely Carson Mays, Jeremy S. Webb, Peter J. Schiff, Shi Yin Fang, Angit Pinician, Suhelen Egan, Peter Steinberg, and lastly, Stefan Schilberg. Um, this well thought out article for me was so well fed, and it provided a no recall type of thesis. So I divided these processes into three main plots to to so we can fully concentrate on their own thesis. I'm not going to dig much deeper on their biological components like um, the the materials that was used and the grams of each each component that they used to achieve this type of concentration or to achieve this type of temperature. Rather, I'm just going to give you guys a gist on what they did and how they accomplished their own thesis. Moving on to their idea, an emerging hypothesis through which C cell bacteria organized as biofilms, biofilms as we all know are bacteria as stated earlier, um, resting in C cell organisms or environments. To be more precise, for example, marine biofilms may exist on abandoned boats, logs in the waters, and even on the top of shells. Okay. Um, an emergent hypothesis through which C-cell bacteria organized as biofilms are turned refuge to avoid predation. The surrounding thought became their very focus, marine biofilms. It was first characterized by the adaptation boundaries of bacteria to deter their consumers. Uh, they they named it secondary metabolites, which are actually neither associated by sex nor growth, unlike metabolites are, unlike the metabolites, unlike the first metabolites. Rather, they act as a toxin to the consumers, indicating stress, to deter these bacterivorous ones or eukaryotic ones. Moving on to their focal point. They drew power from this lack of study, the chemical defense by from bacteria against bacterivorous ones, right? To give further meaning to their idea, they pinpointed the presence of marine bacteria in sea cell organisms and their commonalities in marine ecosystems. Uh, their first method was a method of comparison, um, because what's actually more enticing about their plan was how they are so careful in deciding in having contingencies in in no matter how small the percentage is, they always consider the fact of this of a sample error or a random error. The first method of comparison uh, was they discussed how the high density of C-cell organisms affect the biofilms that are taking refuge. Its difference in concentration from C cells where a group of cells are glued together by an axonometric matrix or planktonic cells. This was made as to differentiate or even acknowledge the difference from the well prevalence of biofilms in adhering a protection against predation than in planktonic species. I'm not going to ponder much on the concentration, but based from their study, planktonic species living in high density areas are typically 2 to 3 times lower than biofilms. Other studies stated include that level of consumers and its mortality rate to both the planktonic species and the biofilms was also concluded, so to further give value to their own thesis, just in case that their own experiment would go array. So they characterize these types of points that may or may not happen in the future. So these are how biofilms forms in the sea. Bacterial cells settles into hard surfaces. Cells proliferate and secrete slime, forming a biofilm layer. Some cells detach, through which it goes on to other sessile organisms or environments. Um, if enough biofilm forms, large organisms can attach to it, like barnacles larvae, as stated earlier. These are the ones that I've stated earlier, through which it can be present on a ship's hull or even an abandoned ship in the water. So yeah. Materials used to justify the results somehow prove to be sufficient and well thought out, as to confidently indicate an average result. The precise thought by staring with the stains or the conditions of each materials used, the growth of each bacteria that was maintained at a certain temperature. So, to further describe it, each materials were met with a certain temperature, um, as they are grown or taken care of, to provide a more suitable or a more precise result. The growth of each bacteria was maintained as stated to avoid any random errors. This was the growth will be mentioned from the three protozoan representatives. I actually forgot. From the three protozoan predators. Um, they were taken care of at a certain temperatures. So yeah. The bacteria that was the focal point uh, uh, was actually dedicated, where they dedicated their own studies to help to help the species in secreting secondary metabolites. But the bacteria that was the focal point is now at phase two. It studies and graces what we call as bioassay, the measurement to define the concentration and the potency of a particular substance. Um, this particular substance was the secondary metabolites. Um, uh, their second phase after the caring of the three protozoan representatives or consumers, um, the marine biofilms and the cells were now their focal point, through which they use it by diluting or minimizing the concentration, through which biofilms kasi are suited in high densities rather than plantonic ones, as I've stated earlier. They use it in a carbon-reduced VNSS medium. I'm not going to ponder on the grams of peptone or yeast or even glucose. But they use those three ingredients to form a bioassay. For the comparison of the plankton versus biofilm's persistence, in a shorter term, it was used to guarantee the persistence of bacteria at given concentration via dilution. Next was phase 4. Phase 3 was the dilution part, was the comparison of the two, their own persistence after minimizing the concentration by diluting. Phase 4 was the production, was I think the production. Um, Phase 4 was the production uh, of analytical capabilities of antiprotozoal activities by focusing more on the volacin production by means of dilution at certain concentrations. Violacin are substances present in secondary metabolites which are used to deter the consumers. They repeated it 2 to 3 times, I think, and as they get the mean and average. The structural integrity through which it was built gave a precise look on the biofilm's persistence rather than the planktonic ones. Thus, it is concludable that the samples made are big enough to be justified at a certain place. Their adaptation qualities may differ from place to place, but each species were thoroughly selected to give their best results without any biases involved. So, those are the causal points. Moving on to the execution. This was actually part of the execution, but still, it was showed that it was actually showed at first that they were actually right on the contingencies. They actually foresee this type of errors. So they actually provide the contingencies, which is actually a plus factor in any thesis or in any articles involved. To always consider contingencies, to always adhere to errors, even though it's at minimum percentage. Um, the results showing grazing resistance of both planktonic species and biofilms were plastered in a quantitative data, through which each result was shown to be highly dependent through bacterial population, as stated earlier, the amount of concentration, because as I've stated, it may differ from place to place. Um, the effectiveness depending on the predator viability and the concentration of the biofilms based from the three protozoan representatives. Um, because of this, they actually have contingencies, and so they adjusted to suit the needs and they back up their thesis through analytical study of each biological components. To summarize, each cell was met to decipher, to show that the conditions met where the violation's defense proved strong, as pure violation was exposed to micro or even submolecular concentrations of other pure protozoan pure violations. Protozoan cells showed subtle morphological changes that are indicative of stress. So basically, it is stating that the violation would prove to be highly effective to the defense, to defending themselves from eukaryotic cells, indicating stress, um, or quickly avoiding the biofilms, as eukaryotic cells approach it within a certain distance. So yeah, so the references for me was a key factor of the research, um, the RRLs and everything. Um, for me, it is a winning factor. The article's references prove to be present in almost any study. They actually have done the research in improving the volatile defenses of marine biofilms in evading eukaryotic predation. Um, this it gave me a well thought of manner on how adaptation really works on marine biofilms, even at sea cell organisms who aren't even moving, um, have evolved of to this type of environments, so they can thrive and survive. I think what's a good factor was how each biofilms, how each critical thinking, sorry, how each critical thinker um, met each error with contingencies. So overall, it was a well thought out article.